Hello. There we go. Okay. Are you working now? Okay. The lid gets stuck. There we go. Okay, we're going to go ahead and get started this morning. So the first thing that I wanted to do is, this is from the UW Advising for folks who are interested in occupational therapy, physical therapy, or prosthetics, orthotics, there's an open house November 12th, Tuesday at 5 p.m. with a little map. Okay? So curriculum information. So this is about going to graduate school. Okay? So you can go ahead and write this down. I can give you a copy. Nick, Fela actually asked me to give this to you since you were just there for advising. Does anyone else need this? Okay, so I will have to post it electronically then because I didn't bring that many copies of it. Okay? So I will post it electronically in Canvas. Okay? All right. So we want to go ahead with the finish our Down syndrome clicker case that we had started last time. So go ahead and find your clickers. And I think that we still need to register 2 people's clickers because I don't think I had you in the roster last time, Nick and John. So let's go ahead and do that. Maybe, if the computer will let us. Okay, so this one I'm not collecting. This one. The spinning blue circle. I'm not sure why it does that. I don't. Why is it still spinning? I don't. The circle is still spinning. Okay, I'm not really sure what's going on with that. I think we should just go back to the chalkboard. All right, so I'm not sure why this won't work. All right. So I think we're going to just try this one more time. All right, so let's just go see if it'll just let us even. So here was our next question. So we were talking about the fact that these folks, the baby had trisomy 21. So you're trying to figure out which of the following is a normal gamete that might be produced by either John or Jane. Let's see if this works. It doesn't matter. Okay, so remember that you're answering this silently without discussing it. All right, so I'm going to go ahead and close the question. Okay, so now you can chat with your neighbor. And remember to discuss your reason or your evidence for choosing a certain one. Okay, I'm going to reopen the question. Okay, so I think that's everybody. I'm going to close the question. Okay, so we have a 60-40 split here. So some folks chose D and some folks chose C. So let's hear from someone who chose C. Yes, Stephen.
I chose C because in both parent you get one of the chromosomes from both parent 1 and parent 2 in C.
Okay, so let's look at the question. It says, which of the following is a normal gamete? So what's a gamete mean? A sex cell, which is a what? Haploid. So it's an egg or a sperm. So the right answer would be D. Yep. Well, it says specifically gamete, either John or Jane. Okay? Gamete, and the word is gamete. So when the egg and the sperm come together, what do we call that cell when the egg and the sperm come together? Zygote. It didn't ask you about the zygote. It asked you about the gamete. Okay? So the correct choice is D. All right. So what went wrong? So you've explained to the jury how cells normally receive the correct number of chromosomes. You need to explain whether this did not happen with John and Jane's baby because of something the clinic doctors may have done. I think that's supposed to be plural and not a possessive apostrophe there. What can cause a cell to inherit the incorrect number of chromosomes? All right. So we have normal meiosis and we have what's called non-disjunction. And essentially with non-disjunction, we skipped a slide. All right, let's go with this. Here we go. Okay, it's time to answer the question. All right, you can go ahead and chat with your neighbor. That's an excellent question, and we're going to get to that. All right, I'm going to reopen the question. Okay, we need one more person to answer the question. I'm going to close the question. Someone hasn't clicked in. All right, so the good news is the second time around, 100% of you chose A. So the chromosomes are not separating correctly during either mitosis or meiosis. So here's normal meiosis, and here's an example of non-disjunction. Okay, so you end up with extra chromosomes, and this would be something that could cause Down syndrome potentially. And here you have an absence of chromosomes. Generally, when there's an absence of chromosomes, if this usually the embryo does not develop, so it dies. Generally speaking.
That would be like a miscarriage, or would you not even really know?
It depends on at what point. It could be a miscarriage. It could happen before you knew you were pregnant. Depends on what cell stage it was at. All right. So this is a similar picture from your textbook showing you non-disjunction. Okay. So it says only one pair of homologous chromosomes is emphasized. In humans, of course, there are 22 other pairs. So this is showing you that it happened in anaphase of meiosis 1. Is that the question you were just asking me? Okay. And this is a link that will takes us to a little animation of this. So it's showing you that it can happen either at meiosis 1 or meiosis 2. Okay? All right, so you can have, so now it's showing fertilization. All right, so trisomy, you can get trisomy from non-disjunction at either meiosis 1 or meiosis 2. Okay? And the other link is actually interesting. This one actually shows photography. So actual chromosomes instead of cartoons. If it works on my computer in my office. Okay, so I guess I have to call media services and ask them why QuickTime doesn't work on this computer. So from the Canvas PowerPoint, you can go to that link, and it's nice because you can actually see the chromosomes. And we'll have to get QuickTime fixed on this machine. All right, so here it's showing you the different copies of chromosome 21 for John, Jane, and their baby are shown here. So we have parent 1 and parent 2. So they're not blaming the parents, right? They're just showing you that the child ended up with an extra copy. All right, so here's your question. When and where did the mistake occur? Remember that you're going to answer this silently on your own the first time through. Okay, so go ahead and chat with your neighbor. Okay, I'm going to reopen the question. Okay, I need one more person to answer the question. I'm going to close the question. Everyone make sure you're oh, there we go, 42. All right. So we have a 50-50 split here. So half of you said meiosis 2, and half of you said either meiosis 1 or meiosis 2. Okay? So how do you know if this is from meiosis 1 or meiosis 2? What evidence would you use? What evidence would you use?
Well, when he and I were talking about it, when you showed all the ones where it's meiosis 1, it has 3 different colors in there.
Okay.
As where with meiosis 2, you would have 2 of one color, and then the parent number 2 would give their color.
So you chose C?
Yeah.
Okay, so what does the color mean? The color of the chromosome, what does that represent? So she's using the color as evidence of where the issue occurred.
That, like, the 2 pink ones were a duplicate of 1, and so that means that they were, like, already split from all of these pairs.
So what do we call the 2 pink ones?
Chromatids.
They were sister chromatids, right?
Yeah.
Okay, they didn't get separated. So the right choice here is C. So it's meiosis 2. So you can contrast this picture as, what's your name again? Serena said it's the color. So in the little animation we had colored chromosomes, and in your book, if I can get the light. So in your book, here, this is page 224. So the book is showing it at meiosis 1. And again, as Serena said, we have 3 different colored chromosomes down here. Okay, so the color of those chromosomes is your evidence. So they were sister chromatids. Yes. Sure. And also the link will be in the PowerPoint. The PowerPoints are in Canvas. I was going to say Angel, but that's not right. Then they'll make us change again. Stephen. Okay? Is that helpful? All right. Okay, so we did. All right, and This is just another slide showing you putting them both on the same page. So here's non-disjunction. This one doesn't have the colors, right? So it's not quite as helpful, but it's showing you non-disjunction meiosis 1 and non-disjunction meiosis 2. Okay? All right, so now you're the jury. What would you decide? That's not evidence because it's right. All right, I need one more person to answer the question. Okay, so the right choice here is B. The doctor had nothing to do with this. Chris?
What happened if two trisomy babies?
Probably the embryo wouldn't develop. So trisomy 21 is one of the few chromosomal abnormalities where the embryo comes to term. Most of the time, if you have missing chromosomes or extra chromosomes, at some point in the development, the developing organism dies. You had a question?
Yeah, could this be avoided in the lab? Like if we just use an in vitro fertilization
For everybody?
for everyone, and just said that we're going to make sure that we have the proper number of chromosomes.
Potentially, but then the expense of that would be crazy, and a lot of people have ethical issues with that as well. So potentially yes. If they do in vitro, sometimes they are doing tests. It really depends on the doctor and the parents and what their procedure is. So, and some people do in vitro if they have a genetic, like Tay-Sachs disease, which genetically the kids die a awful death by the time they're 4 or 5 years old. And if the people both know they're carriers of that, they might do in vitro and they might pre-select the embryos to be implanted. But some people are okay with that, and other people have ethical concerns with that. So, all right, so case closed. You presented pretty conclusive evidence that the clinic was not responsible for causing Down syndrome in the baby. However, should the clinic have alerted the couple that something might be wrong before the baby was delivered? How could the doctors have known that the baby might be born with Down syndrome? All right. So there are tests that you can do for this, and this is called a karyotype, and this will detect gross chromosomal abnormalities like extra chromosomes or missing chromosomes. Okay, so this is a standard type of a test that you could do for Down syndrome, and they can do this through amniocentesis, or they can do chorionic villi sampling. So they have to get some fetal cells to do this type of a test. Okay? So that's karyotyping. And somewhere in your textbook, oh, it's right here, page 225, you have a picture of that. All right, so when would be, this is your question that you're going to answer silently, when would be the best time to isolate chromosomes for a karyotype? Okay, let's go ahead and answer the question, and then you'll have a chance to chat with your neighbor. So 2 more people need to choose. I'm going to close the question. All right, go ahead and chat with your neighbor. All right, I'm going to reopen the question. Okay, I need 2 more folks to click in. Okay, so we are sort of all over the map here. All right, so no one picked A. All right, so we have S, G2, prophase, and metaphase. Okay, so let's have someone who chose B talk about why you chose S phase. Why did you choose S phase? 9 people chose S phase.
I chose S phase because S phase is when the DNA replicates.
Okay.
If you're going to isolate, it would be good to wait until after the DNA was replicated to find, would that lead for, more chance of, finding the problem?
Okay, so you chose S phase because that's when the DNA replicates. So that means you didn't choose G1, but if it's already happened and using your evidence, doesn't that make all these equal? S, G2, because it's duplicated in all of those? Yeah, it does. Okay, so if you chose S because of synthesis, so that only rules out G1. Okay, so what else happens to make one of these other things a better choice? Kelsey.
I chose metaphase because crossing over has already happened, so the DNA is already how it will be when meiosis 2 happens.
Okay, so is this meiosis or mitosis we're talking about? So this is the developing fetus. Mitosis. Meiosis only makes the egg and sperm. Once the egg and sperm come together, you have a zygote. Everything after that is going to be mitosis. So crossing over is not relevant because that's not happening during mitosis. Okay, someone from this side of the room. What else happens? So the DNA duplicates. That's important. What else happens with the chromosomes during mitosis? What do they do? Yes, in the back. They're all lined up. That's good. They're all lined up, so it's going to be easy to see in the cell what's going on. What else has happened between the beginning, G1, and metaphase? It's going to make it easy to see what's going on at metaphase. They're separated. They're also what? Condensed. They're condensed. The chromosomes are all packed up and supercoiled, and you can see that they're separate chromosomes. If you were looking at G1, S, or G2, they're all loose and thready. They're not condensed. Okay? So in metaphase, they're all sort of neatly lined up there in the middle. So that's going to be the best choice. The nuclear membrane is going to be gone as well. So they're going to be a little bit more spread out in the cell. Okay? So no. All right. So here, this one should be easy. Does this person have Down syndrome? Answer this silently, please. And I don't want you guys using your ebook to answer the clicker questions, okay? I'm expecting 100% on this one, guys. Don't let me down. Stephen automatically takes the... All right. I'm going to close the question. Yes. How do you know this person has Down syndrome? 1, 2, 3. All you have to do is count the little chromosomes. Okay? So this is a karyotype. It's not colored, but it's just a black and white karyotype. Yes, you have Down syndrome. So Down syndrome can be more or less severe,
Okay, with that, okay.
but if you have 3 copies of this, you have Down syndrome. Yes. All right. So karyotype can be performed by collecting cells from the developing embryo. The techniques, chorionic villi sampling and amniocentesis, do slightly increase the risk of miscarriage. Jane's family had no history of Down syndrome. John had a cousin with the disorder. It is related to maternal age, and that's been known for quite some time. There's newer research. It can also be related to the paternal age, the age of the father. But
how does, genetics, how would that affect?
a lot of it's just random. So why is it maternal age? Because the eggs are older. We don't really know.
But, genetically, since it said his cousin had the disorder, that doesn't really affect it, does it?
Not typically. No. All right. Is the clinic liable for not testing for Down syndrome? Again, you're just answering this silently. Okay, I need one more person to answer the question. Okay, so this is essentially an opinion question. There's not a correct answer for this. Okay? And various courts have decided this differently. So in France, the Court of Appeals ruled that the parents should be compensated because the situation was not detected by the doctor during pregnancy. This, I don't know. So it is an invasive procedure. There are some signs that can show up on some ultrasounds. There can be some abnormalities of the heart, etc., that sometimes can be noticed on an ultrasound. But the definitive test is this invasive procedure. Yes, Tom?
Well, what would testing for detecting it do?
You can't prevent it. You would choose to have the baby or you would choose to terminate the pregnancy. Those would be your only options. Some people, they kind of want to know, perhaps, to be psychologically prepared, but there's no way to treat it. It's not a treatable condition. In 2004, the Supreme Court said that they could not sue the doctor for misreading the test. So if they did the test and the doctor misread it, I would have a little more issue with that on the jury myself. But as we said, the courts have decided this differently. And here in 2007, the medical center failed to offer her the opportunity.
That's different.
So these are all, different court rulings, and different folks are going to have different ideas. That's why you are paying the lawyer to argue your case. Okay. Historically, women over age 35 were encouraged to have either amniocentesis or chorionic villi sampling, but most pregnancies occur in younger people. So 80% of Down syndrome cases occur if you're under 35. Okay? So just because that's not a guarantee that you're going to have it or not have it. Okay? You have to do the test to know. In 2007, the American College of OBGYN professionals recommended that every pregnant woman be offered a choice of the test. Okay? So, and if you want more information, there's more at this Down Syndrome Center. Okay, so that essentially concludes the mitosis and meiosis coverage that we're doing. Some of you had lab yesterday. The rest of you have lab tomorrow. So you'll be seeing us again in lab. Okay? Any questions about what we covered here with mitosis and meiosis? Okay, so we are going to go ahead and move on to Mendel and Punnett squares. So we're and one of the goals, and I talked about this in lab yesterday, is for you to combine your understanding of what's going on with meiosis to Mendelian genetics. So it's essentially the behavior of the chromosomes during meiosis explains what Mendel discovered. And the fact that Mendel discovered it without using a microscope, ever seeing a chromosome, is actually pretty amazing. All right? So we are going to focus on this chapter 12 as the chromosome theory of inheritance, which is a phrase that may or may not be used in your textbook. I'm not sure. All right. So we need to make sure that we understand some of the terminology, and some of this is in your labs and pre-lab stuff. Allele is essentially a different form of a gene. Okay? Homozygous means you have the two copies that are the same. So if you're little s, you're homozygous. So the small lowercase letter indicates a recessive trait or a recessive allele, right? So recessive, and again, these are conventions, equals lowercase. And a capital letter equals a dominant trait. Heterozygous means they have one copy of each, a big S and a small s. Okay? So Mendel's first law is the law of segregation. The two copies of the gene, and he didn't use the word gene. Okay, so the way they're presented in the text has been updated with modern language. So he talked about it as the particulate theory of inheritance. The two copies of a gene separate when an individual makes gametes. That's his first law. So we now know that this is explained by the homologous chromosomes. So the homologous chromosomes bear the two alleles for each characteristic. And we now know that his law is explained by the separation of homologous pairs during meiosis I. And so this is a figure, this one I have borrowed from another textbook because I like it and there's not something similar in your book. So this shows you homozygous for the dominant allele, homozygous for the recessive allele, heterozygous. So you have big P here, you have little a here, and you have a big B and a little b here. So the other word that you want to know here is locus. And locus, so the plural of locus is loci, and that essentially means its physical location on the chromosome. So one chromosome actually can have hundreds of different alleles. And we're just showing you 3 here. But they need to physically be in the same place on the chromosome. So yesterday you were playing with little pieces of Lego, and I said, oh no, you need to keep those in the same place. Why do you need to keep them in the same place when you're playing with Legos? Or why does a cell need to keep them in the same place? Patrick?
Because, like, if they're loci,
Loci.
they have to be located in the same place, otherwise they're different alleles.
So what would happen if they weren't in the right? So what if on this chromosome these were switched? And when would that become a problem? You can sometimes have normal protein synthesis if they're in a different order. You can. Depends. But when in the process of mitosis or meiosis would this be a problem? Crossing over. You would get 2. So if these were switched and you had a crossover event here, you would have 2 copies of the allele on one chromosome and no copies on the other chromosome. Okay? So this would be a problem for crossing over of meiosis if they weren't in the same physical location. Yes? Are these sister chromatids? That's a good question. No. Why not? They're homologous chromosomes, okay? They're homologous. They're not sister because they have different alleles, right? So here we have a big B and a little b. And they've also they've color-coded them. But those are homologous. So has S occurred yet? No, they haven't replicated themselves yet. So in your normal cell, if it's not undergoing cell division, they actually aren't condensed either. We've shown them here in their condensed form so you can see them. But for example, this would be the one that you got from your dad, and this would be the homolog that you got from your mom. So they have the same genes, but they have different versions of those genes. So those are homologous chromosomes. They're not sister chromatids. Okay? And that's why I like this figure, because it helps explain those relationships, which are important to understand. All right. So this is the picture from your book. I think the next, I have made it bigger. Okay, so we're looking at the top half of this figure. So here are the homologous chromosomes. So we have a big S and a small s. This is showing you meiotic interphase. So now S has occurred, so now they've duplicated. And then here we have at the end of meiosis 2, we have 4 haploid gametes. And we've only shown 1 chromosome, and so we didn't do crossing over here. Okay? All right, so now let's convert this into the Mendelian stuff. Find your carbonless paper, and if you were crossing this gamete and this gamete, what would your Punnett square look like? So one is big S. Okay, I'm going to let you guys figure that out. So you're going to do a Punnett square, and you're going to cross one of the red gametes with one of the blue gametes. Actually, you're going to cross this with that. Sorry. I know. I figured that out as I was saying it. I didn't say it right. Yes. So you're doing a cross of 2 homozygous. You meant homozygous, not homologous. I knew that. Okay, you guys need to get something written down on your paper. Okay, who wants to be brave and let the class look at your paper? Tajana. Thank you. She loves doing Punnett squares. Okay, that's good. I think so. Best part of high school biology. Okay, so you should have something that looks like this. Big S, little s, little s. You can have them flipped either way. Yes, if you use cursive, it's easier to keep your big S's straight from your little s's. Okay? All right. So good job, Tajana. Okay, so what do we call the big S across the top here? In terms of, they're homozygous, and this is the parent. This is P, right? And this is P. P means parent. So what do we call these things in the middle? F1. So these are F1. Okay, so which means first filial, which is the first generation. So now what I want you to do is on your same paper is now cross the F1s.
Do we need the pheno-geno thing?
I didn't ask you to do that.
Just 2 of the F1s?
So, yes, 2 of the F1s. So now you're going to cross 2 of the F1s. Yes. They should all be the same, so the results would be the same regardless of how many you cross. If that helps you, sure. Or you can just make little cursive S's. Okay, who wants to share their paper? All right, so now you should have something that looks like this. So you crossed big S, little s, big S, little s, and you have big S, big S, big S, little s, big S, little s, little s, little s. Awesome. Okay, so what do we call the organisms in here? F2. Okay, so those are the F2. All right, so what do you notice about the F2 generation? What can you say about the F2 generation? So you want to talk about the genotype ratio and the phenotype ratio. So what would be the phenotype ratio? 3 to 1. Okay, so the phenotype is the physical appearance. So the phenotypic ratio would be 3 to 1. So 3 of them are going to exhibit the dominant trait, and only 1's going to exhibit the recessive trait. What about the genotype ratio? 1 to 2 to 1. Okay? So you had 1 big S, you had 1 big S, little s. Sorry, you had 2 of those. And you had 1 little s, little s. Okay? All right, so if you had lab on Tuesday, so at the end of your chromosome packet, there was the crossing, which some of you started working on. So those, you want to have those done for Friday. So you're going to move on to the dihybrid cross. Okay? And you do have animations in the bioportal to go through if you're not sure how to do that, and you can work on that in lab tomorrow if you haven't had lab yet. So any questions about Mendel at this point? All right, go ahead and just put those back in your, go ahead and keep your carbonless paper today. Okay? You do want to move on to Mendel's second law of independent assortment. You want to make sure you understand the 2 laws. So we do have the problem session at 11. You have 10 minutes to go take a break.